Hello everyone, welcome to ITLS Academy. ITLS Academy is the best online training platform with unique features which you will not find anywhere else. I am Siddharth Singh, MTech from IIT BHU and expert in cancer biology. So today's lecture will be on tumor immunology. So let's start. So we will start with the introduction of what is cancer and what is a tumor and how these two terms are related, or is there any similarity between these two terms or not. So what is a cancer? Cancer is a group of diseases which involves abnormal cell growth with the potential to invade or spread to other parts of the body. So basically, a cell, when it starts behaving abnormally or starts dividing abnormally, it becomes a cancer. Normally it might be the case that one cell needs to divide only one time, but due to development of cancer or due to the disease of cancer, it is now dividing 100 times. So it is producing much more cells which are required for the human body to work. So that function or that part of the cell part, that tissue part will show abnormal growth. So this is cancer. Now what is a tumor? Tumor is a swelling of a part of the body, generally without inflammation, which is caused by an abnormal growth of tissue, whether benign or malignant. Basically, tumor is the abnormal growth but without inflammation. So now, how these two terms are related, cancer and tumor? We can say that every cancer is a tumor, but every tumor is not cancer. Because cancer is having the capability to spread throughout the body or throughout the different organs, whether tumor confined to its own place. It doesn't migrate to other places. For example, if a person is having kidney cancer, then there is a high possibility that these cancer cells can migrate to other organs, to the nearby organs, like for spleen, for example, and these cells can start developing or start cell division there, and then they start developing cancer there. And then the person is now showing a spleen cancer. Whereas in the tumor, if a particular organ or a particular tissue is showing tumor, then there is high chances that it will only confine to this tissue or this organ. It will not migrate to other tissues. Let's move further. So how does cancer arises? We can say that all cancer begins in cell. Our bodies are made up of more than 100 million cells or a billion cells. Cancer starts with a change in one cell or a small group of cells. For cancer to start, certain changes takes place within the gene of a cell. So basically, if you focus on this diagram here, this is the diagram of a cell showing different organelles and various other components of the cell, like ribosome, centriole, lysosome, cytoplasm. All the organelles has been shown correctly in this diagram. So what cell changes and cancer means? It means that all the cancer starts developing from a single cell. Any of the cell of this or of human body or suppose or any other organism will show some changes, or a group of cells can show some changes, which will start gathering they will start gathering, and after some particular set of time or after a particular amount of time, then they will show the phenotypic results, like the abnormal cell growth. The abnormal cell growth will be visible only after certain time limit, like suppose 1 month, 2 months. It takes time to show on the surface. Now, coming to the next point, genes and cell division. Now we all know that different types of cell in the body do different jobs, but they are basically similar. They all have a control center called a nucleus. Inside the nucleus are chromosomes made up of thousands of genes. We also know gene contains long strands of DNA which are coded messages that tell the cell how to behave. Each gene is an instruction that tells the cell to make something. This could be a protein or a different type of molecule called RNA. So basically, all the cells, they are having a nucleus. They are inside the nucleus. They are having a chromosome. Then they are having gene. Then all the genes are a set of instruction to code for a particular protein or to code for a particular RNA or some other things which are required in the body. Because 90% of the times the protein is required in the body, so all the genes are the coded messages to form a particular protein. Now coming to the next point, gene changes within a cell, which is called as mutation. So normally genes make sure that cell grow and reproduce in an orderly and controlled fashion. They make sure that all the cell produced are needed to keep the body healthy. But sometimes a change happens in the genes when a cell divides. This is called a mutation. So basically mutation is what? Suppose DNA replication process is happening within the cell. And if suppose the template strand is showing the sequence ATGC, but the strand which is being synthesized by the DNA polymerase, somehow the DNA polymerase is making a mistake of single base. So now, suppose in this sequence the complementary sequence should be T, A, C and G, but because of the DNA polymerase ineffectiveness, this one G is been replaced by a C. So this is what we call as a mutation. Ideally C should always pair with G, but in this case, because of the ineffectiveness or the of DNA polymerase, G is now being replaced by C. So this is called a mutation. Now because of this mutation, whatever the protein is going to be synthesized from this gene further down the line will not synthesize, because there is a mutation in this gene. Some other protein will be synthesized. Now suppose in original case the protein which is being synthesized is for an eye color. Now this mutated gene will be synthesized, or mutated protein will be synthesized, giving some other color or no color at all. So this is what you mean by mutation. So it means that a gene has been damaged or lost or copied twice. Mutation in a particular gene may mean that a cell starts producing too many proteins that trigger a cell to divide, or it stops producing protein that normally tell a cell to stop dividing. Because of mutation, it can also happen that earlier case, in regular fashion the cell cycle is regulating the cell division process, but due to the mutation, those proteins which are giving signals to stop the cell and to start the cell cycle, they are mutated, so the cell is not regulated. So more number of cells can be synthesized, or they can be, there is no check on the cell division of a cell cycle. So in this diagram you can understand a difference between a normal cell and a cancerous cell. Like you can see the cytoplasm, the nucleus, the normal cell is only having single nucleus, while the cancer cell is having multiple nucleuses. Same wise with nucleolus. A normal cell will have only single nucleolus, but a cancer cell will have a multiple nucleus. Same wise chromatin. You can see the difference between the chromatin in normal cell. It is very very lightly represented, or very lightly thread-like structures, but in cancer cell they are very thick structures. Cytoplasm also get reduced. In the normal cells we have a good amount of cytoplasm as compared to the cytoplasm which is present in cancer cell. The volume of cytoplasms get reduced in the cancer cell. Now moving on to the next slide, what are the concepts in tumor immunity? So basically if we try to understand with the help of a diagram, then you can see that this is a tumor cell, and we are having tumor antigens, various tumor antigens. These might be protein. So now the tumor immunity is a process which our human body is capable of generating against a tumor cell, because all of us are not having cancer cells, or all of us are not developing cancer because of our immune system, because of our immune capacity, that we are killing these tumor cells. So what are those mechanisms which are in place to help us beat the cancer? So let's suppose this is the tumor cell. So we are having humoral response and antibody mediated response. So B cells and T cells will be there for the killing of tumor cells. So in this pathway, these antigens will be carried out, or these will be represented, or will be tackled, or will be handled by the B cells here. Then B cells will generate their antibodies. We all know that B cells generate antibodies and the memory cells. So these antibodies will be generated, and these antibody will kill the tumor cell. Another method is these antigens can be internalized by antigen processing cells or dendritic cells, which will engulf these peptides or these tumor antigens, and they will represent it on its surface via the MHC2 complex to the T helper cells. T helper cells, we all know that cytotoxic mediated cell toxicity, we have TC cells, we have TC cells, and we have TH cells. TH cells are T helper cells and TC cells are cytotoxic T cells. So T helper cells, they help in generating or activating the TC cells via the process of cytokines. They also mediate the killing of cells, but not to that extent. Main responsibility of killing any foreign antigen or any foreign pathogen is on the shoulder of TC cells. So TH is basically for the activation of TC cells. So in this process, the MSC bound antigen or the MSC bound tumor peptide will be recognized by TH cells, and they will secrete cytokines will in turn help in the activation of TC cells. Now TC cells, what they will do? They will interact with the T cell receptors and with the MHC1 complex with the tumor cell. So now once this tumor cell on MHC1 class complex is formed, these cells mediated or initiate the cell mediated cytotoxicity. By this process, TC cells can kill the tumor cell. Other cells which are in play or which plays a role in the killing of tumor cells are natural killer cells or macrophages. In natural killer cells, when there is a reduced expression of class 1 MHC expression or class 1 MHC molecule, basically there is a reduced synthesis of TC cells, then the NK cells plays a role. They plays a role when there is a reduced class 1 MHC expression and expresses ligand for NK cells which are activating receptors. So basically, when TC cells are not synthesized in efficient quantity or appropriate amount, then natural killer cells can also mediate the killing of a tumor cell. Other cells which can play a role in tumor killing are macrophages. Macrophages are basically, these are the first cells which respond to tumor cell infection. So basically these also can kill the tumor cells. Now tumor specific antibodies, I have already discussed the B cells. They will generate tumor specific antibodies and will mediate this antibody dependent cell cytotoxicity. ADCC is the short form of antibody dependent cell cytotoxicity. So this is the complete process, how after a generation or after the induction of a tumor cell or after a generation of a tumor cell in human body, how our immune system functions to kill that tumor cell. Basically a few points, just a recap of this whole process. And tumor antigens will be processed by B cells, either by B cells or T cells, or by natural killer cells, or by macrophages, or by tumor specific antibodies. So let's move further. Now this is the cancer immunity cycle. So what do I mean by cancer immunity cycle? It is a complete process by the induction or by the initiation of cancer, and how the complete process takes place till the killing of the cancer cell. So first step is the release of cancer cell antigens. After the cancer's death, there is a particular cell which is showing abnormal cell growth. Suppose it is getting internalized or it is getting killed by some immune system. Now what happens? It will release other cancer cell antigens. Now these cancer cell antigens will be diagnosed or will be captured by antigen processing cells. In our case, mostly these are dendritic cells. So they will be represented by these APC cell, antigen presenting cells. So second step would be cancer antigen presentation via dendritic cells. Now next step would be the priming and activation of these cells. Now these cells, after they engulf or after they attach themselves to the antigen, they prime those antigen so that they can be efficiently killed by the TC cells or the further processes. So third step would be priming and activation of T cells. Now the fourth step will be trafficking of T cells to tumors. Now once the antigen is being prepared for the killing, now the T cells will be moved towards those antigen presenting cells, CTLs. Now in fifth step will be the infiltration of T cells into tumors. So now the the TC cells will migrate towards the antigen presenting cells. Now they will reach further close to them via the blood vessels. They will reach towards those antigen presenting cells. Now the sixth step would be the recognition of cancer cell by T cell. This will happen through T cell receptor, which we call as TCR. This TCR will bind to the cancerous cells receptor and will recognize that it is a cancer cell. Now after this binding, this will initiate the process of killing of a cancer cell via the immune cells. So the seventh step is finally the killing of the tumor cells. On the next slide, we will understand what is a tumor immunology. So basically, tumor immunology means the role of immune system in the progression and development of cancer, or the study of the response of immune system to cancer. Earlier we have seen that what all the cell molecules or the cell which are involved in the killing of a tumor cell. In the tumor immunology, we'll understand the study of a response of immune system to cancer. So basically it is the same thing, just it is a name given to the science or the study of those molecules, how they are killing or how they are interacting with the cancer cells in order to excrete those dangerous cells from our system. So what are the goals of tumor immunology? Immunology, the first goal would be to describe the different types of tumor antigens, because in the previous slide I have told you that the tumor cell will release tumor antigens, whether they can be peptide or they are some other molecules. We will understand what are these molecules. So the first goal of tumor immunology is to understand what are tumor antigens. Next goal would be to understand how the immune response fail to allow tumors to grow. Now, because we all know that all of us don't develop cancer, but some of human beings, they are developing cancer, which means that there is a loophole in our immune system, or there is a malfunctioning of our immune system somewhere throughout the killing of a cancer cell, which we will try to understand where are those loopholes present and what we can do to avoid those loopholes. Now the next goal would be to understand how we can use the immune response to fight cancer. So what are the protective mechanisms, or what are the medicines, or what are the therapies which are available in the current world to efficiently respond to the cancer cells. So let's start with the tumor antigen. So tumor antigen is an antigenic substances. We all know antigen will be the same what the antigen definition in normal immunology is. Antigen is any foreign particle which enters in the human body. So tumor antigen is an antigenic substance produced in tumor cells. That is, it triggers an immune response in the host. Tumor antigens are useful tumor markers in identifying tumor cells with diagnostic tests and are potential candidate for use in cancer therapy. So tumors may express tumor antigens that are recognized by the immune system and may induce an immune response. These tumor antigens are basically tumor antigens can be divided into two categories. One is tumor specific antigens and another one is tumor associated antigens. So what is the difference between these two antigens, or these two types of antigens? The tumor specific antigens, or TSA, are antigens that are only present on the tumor cells, and tumor associated antigens, or TAA, are present in healthy cells, but they are also present on the tumor cells. So tumor specific will only be present on the tumor cell, and tumor associated antigens will be present on both the cells, both healthy cells and the tumor cells. Let's move further. So in this slide we will try to understand what is immune surveillance, or what our immune system does once it recognizes the cell that it is a cancer cell. So let's start with the definition of immune surveillance. So immunological surveillance is a monitoring process of immune system to identify cancer and or precancerous cells and eliminates them before they can cause harm. So the process of immune surveillance is to constantly check to identify cancerous and precancerous cells and eliminate them before they can cause any harm. So basically we have to remember in immune surveillance that there are 3 E's. One is elimination, another one is equilibrium, and another one is escape. So let's try to understand with the help of this diagram. that what all we are trying to explain in this diagram. So let's see, this is a normal, these are normal cells and these are transformed cells. So what is happening in throughout this process, that normal cells are becoming cancerous cells. So carcinogen, the exposure of carcinogen or chronic inflammation, inherited genetic mutation, radiation, viral infection, these are all the carcinogen or mutagens which can cause mutation in normal cells to promote transformation of cell to develop into cancerous cell. Some of them they can repair, like because of certain mutation there is a DNA mismatch repairs are present that which can repair the mutation caused in the DNA replication. So if there is a repair process present, then these normal cells, while in the process of becoming transformed cells, can revert back to normal cells if there is a repair process present, senescence is present, or apoptosis is the killing, that the human immune system will recognize that these normal cells are getting mutated or getting transformed into cancer cells. Then they can kill those cells via the process of apoptosis. Or intrinsic tumor suppressor genes, these are the genes which can help in the reversion of transformed, not the completely transformed cells, but in the cells which are in the process of being developed into cancer cells. So these genes can help in the stopping of cancer. So these are the transformed cells. Now these transformed cells are the final, or we can call them as cancerous cells. Now let's try to understand the concept of immune or cancer immunoediting. So there are three processes after a particular individual develops a cancer, or in the process of developing cancer, like some of the cells are already transformed and some cells are being transformed in a particular individual, then the immune system can work via the three processes: the elimination, equilibrium, or escape. So elimination is the process which we call as immune surveillance. So what it does is, suppose these are the cancerous cells which are developed in this cycle. So via the process of innate and adaptive immunity, these cancer cells can be killed via the various molecules or the various protein like interferon gamma, perforin, trail, interferon alpha and beta, and NKG2D. These molecules can help in the elimination of these cancer cells. So first process is the elimination. So once the cancers develop, then via the process of innate and adaptive immunity, and with the help of these molecules in IFN gamma, IFN alpha and beta, our human body can kill those cells, and hence our normal cells will only remain in the human body. So this is the protection, that is extrinsic tumor suppression. So in the first process we are killing the tumor cells. In the second process is an equilibrium process or cancer persistent. It means that the human body is in balance. Like suppose there are certain cells which develops cancer and human immune system are killing them, and on the other hand new immune cells or new cancer cells are being developed. So there is a cycle that as soon as 10 cells are getting developed into cancer, they are killed by the immune system. Next 10 cells are getting developed into cancer, they are being killed by the immune system. So there is a cycle. As soon as the cancer cells are being developed, they are getting killed by the immune system. But there is a continuous cycle. There is this is a non-ending supply. The cancer cells are constantly being generated and they are being constantly killed by the immune system. So there is an equilibrium, or the generation of cancerous cells and the killing of the cancerous cells is an equilibrium. As soon as the cancer cells is getting developed, they are being killed, and new cells are being developed, new cells are being again killed. So now the third process is escape, cancer progression. We will try to understand this cancer progression or escape in the next few slides. For now we will suffice by saying that this is the process where actual cancer develop in a human being. But like there is an immune system in place, but these cells, they evade these immune system via various pathways. We will try to understand what are those pathways, or what is the process by which the immune system fails to recognize these cells and fails to kill those cells, and hence development of the cancer occurs. So we'll try to understand tumor escape mechanism. So what are the three mechanisms? One is low immunogenicity, another one is antigenic modulation, third one is tumor induced immunosuppression. So what is low immunogenicity means? Basically, low immunogenicity means there is no peptide MHC ligand, no adhesion molecules, and no co-stimulatory signals. All the processes that we have tried to understand, or we have discussed in the previous slide, that once the tumor cell is there will be a tumor antigen, and it will be presented by an MHC cell or MHC1 complex or 2 complex, or by antigen presenting cells. So there is no low immunogenicity means that there is no interaction with the MHC ligand, or there is no adhesion molecules by which the MHC1 molecule can bind to B cells or T cells. There is not enough adhesion, or there is no co-stimulatory signal, like the TH cells will help generating or activating the TC cells. So there is no low or no co-stimulatory signal. So the TH helper cells, they cannot activate the TC cells for the efficient killing of the tumor cells. So basically it means decrease in the MHC expression or failure to produce tumor antigens. Now second one is antigenic modulation. What does it mean? Antigenic modulation, it means basically, as the word itself suggests, the antigen is being modulated to some other form. So basically mutation, there is a mutation can occur in the antigen. Like suppose this is a tumor cell and it is producing some antigen. In normal case, this antigen is of some other structure, and in the tumor escape mechanism, if the tumor cell is applying the tumor escape mechanism, then this antigen can adapt to some other form or some other molecular structure which is not being recognized by our immune system. So basically a mutation can result in structural changes in tumor antigens such that they are not recognized by our immune system or immune cells. Now what is tumor induced immune suppression? Tumor induced immune suppressions, or Treg cells or T regulatory cells, factors, example TGF beta, secreted by tumor cells inhibit T cell directly or kill them. So basically what happens in tumor induced immune suppression is the tumor cell there are T regulatory cells which are already present in our immune system, which are secreting TGF beta. These are the molecules which can help in the killing of tumor cells. So these tumor cells, they help in the reduction of these molecules. As soon as the reduction of these molecules happens, so there won't be an efficient killing of the tumor cells, and hence some cells will get killed and some cells will not get killed, and this is how a tumor can escape. Like suppose 100 molecules of tumor are synthesized, or 100 cells of tumor are being synthesized. Out of them, only 50 cells are getting killed by the molecules which are usually present or kill the tumor cells, like TGF beta. Then the remaining 50 cells can thrive or can again multiply. So this is a tumor escape, how the tumor cells can escape from the regulatory mechanisms which are already present to kill the cells or kill the tumor cells in our human body. Let's move further. So how we can use the immune response to fight tumors. So basically this topic covers what are the current therapies or what are the current treatments which are present for killing the tumor cells. So first one, we will discuss the cancer immunotherapy. As the word suggests, cancer immunotherapy is sometimes also called immuno-oncology. It is the artificial stimulation of the immune system to treat cancer. Like basically we already know that our immune system is in place to kill the tumor cells. So in this therapy, what we do, we artificially stimulate our immune cells or immune system to kill the cancer cells, which results in the improving on the immune system natural ability to fight the disease. So basically we are activating on only the already present immune system which kills the tumor cells to kill the tumor cells. Now the cancer immunotherapy can be divided into two forms. One is an active form and another one is passive form. So what is the difference between an active form and a passive form? Active forms direct the immune system to attack tumor cells by targeting tumor antigens. So basically it directly attacks the tumor cells by targeting the tumor antigens. While in passive, they enhance existing anti-tumor response and include the use of monoclonal antibodies, lymphocytes and cytokines. So basically in this process, we are directly targeting the tumor cells by targeting on the tumor antigen, and in this process we are activating our immune system, or we are indirectly targeting the tumor cells by enhancing our anti-tumor response, by the process of monoclonal antibodies or activating cytokines or lymphocytes. So this is basically this is the main difference between an active and passive cancer immunotherapy. Let's move further. So what is, in detail we can understand from this slide, what is an active immunotherapy. So in active immunotherapy, as I've already told you, that we will target the tumor cells by targeting their tumor antigen. So what in this diagram we can show, or we can understand from this diagram, that blood cells are being removed from the body of a cancer patient. Now these mononuclear cells are incubated with tumor antigens. So now these are the cells which are being removed. So now these are being incubated with the known tumor antigens or the tumor antigens, so that these cells gets activated to the full form, to their fullest form. Once these cells, or these cells by the way are our immune cells. So we have removed the immune cells from our human body, and then we have kept, or we have incubated them with the tumor antigens. So what will happen after the incubation? These cells will get activated and they will start killing these antigens. So these are the fully activated tumor antigen killing cells or immune cells. So once these gets fully activated, then we are again transfusing back towards the human body, or from the patient from which we have taken the blood. So mature dendritic cells cells displaying the antigens and are transfused back to the recipient. So once they are getting fully activated, they are being again transfused back towards in the cancer patient. So one of the drug which is being approved by FDA for this process is Provision. It was approved for the treatment of asymptomatic or minimally symptomatic metastatic castration resistant prostate cancer in 2010. So there are many other drugs which I have not mentioned in this slide, but this is one of the drugs which is actively being used for the process of dendritic cell therapy. Moving further, another therapy is CAR T cell therapy or chimeric antigen receptor therapy. So what we do in this process, we modify T cells to recognize cancer cell in order to more effectively target and destroy them. So scientists, they have harvested T cells from people, genetically alter them to add a chimeric antigen receptor that specifically recognize cancer cell, then infuse the resulting CAR T cells into the patient to attack their tumor. So basically this is the same process, the active process. They have harvested the T cells from the people, then genetically they have altered them to add the CAR, which can specifically recognize cancer cell, and then they have again infused back these resulting CAR T cells into the patient to attack their tumor. So this is the whole process of again the active immunotherapy. This is another example, which is therapy which is already present in the market for the treatment of tumors or cancer cells. Let's move further. So the passive immunotherapy, as I've already told you, that in passive immunotherapy we will only enhance the existing anti-tumor response via the production of monoclonal antibodies or lymphocytes, by adding the lymphocytes or by activating the lymphocytes and cytokine production. So this is all about passive therapy. We will try to understand the process of monoclonal antibodies, how the process of monoclonal antibodies help in attaining this process. So let's try to understand with the help of this diagram how with the help of monoclonal antibodies we can kill the tumor cells. In the first diagram we can see that there is a tumor specific antibody. Now antibodies binds to the tumor cell. We can see that this is a tumor cell, and these are the antibodies which are getting bound to these tumor cells. Now what happens is NK cells or natural killer cells with Fc receptor CD16s are activated to kill these tumor cells. Because of these antibody, these NK cells will bind to these antibody and they will mediate the killing of the tumor cell. Now another method or another process by which monoclonal antibodies can help in the efficient killing of the tumor cell is by generating tumor specific antibody conjugated to a toxin. So we have conjugated, or we have added a toxin molecule with the monoclonal antibody which is very specific to a particular tumor. So now what would happen? Antibody toxin conjugates bind to the tumor cell. Normal process, these antibody will bind to the tumor cells with their toxin conjugate. Now with the help of these conjugates are internalized, and hence the killing of the cell occur. So now the toxins are internalized by the tumor cells, and because of their toxin nature, they will they will mediate the killing of a tumor cell or the cancerous cell. Now the third process in which monoclonal antibodies can help is tumor specific antibody conjugate to a radioisotope. So in this case we have conjugated them with a toxin, and in this case we have conjugated the monoclonal antibodies with a radioactive isotope. So what would happen with the help of radioisotope? Radioactive antibody binds to the tumor cells. These antibody will bind to the tumor cells. Now these radiations will generate a signal. Then radiations kill the tumor cell and neighboring tumor cell. Now because of this radioactive molecule, which is attached to the monoclonal antibody, these radioactive molecule will generate radiations or will produce radiations. Because of those radiations, there is an effective killing of tumor cells. Now here in this list I have jotted down some of the antibodies which are monoclonal antibodies which are produced or which are approved by FDA, and their target and their treatment, in which type of cancer they are being used. So the first antibody is alemtuzumab, which is the target. Target is CD52. CD52 is a marker which is present on the cancer cells. So this antibody is being utilized in the treatment of B cell chronic lymphocytic leukemia. Another one is atezolizumab, which is the target is PD-L1, and the treatment for which it is used is the cancer is bladder cancer. Another one is avelumab, target is same PD-L1. It is being used in the treatment of metastatic Merkel cell carcinoma. Another one is ipilimumab, the target is CTLA4, and it is being used in the treatment of metastatic melanoma. So basically this is the complete process how monoclonal antibody killing of cancer cell can happen. So let's move further. Now another method or another process by which we can mediate the killing, or we can treat tumor cells or cancer cells or cancer or cancer humans individual is a cancer vaccine. So we already know what a vaccine is. So the key element in the design of strategies for vaccination against cancer, which are, first one is the identification of significant tumor antigens by genetic or biochemical approaches. So first of all we have to generate significant tumor antigens, so that against those antigens the antibodies can be made. These vaccines are what? They are generally antibodies, right? So we have to generate or identify the significant tumor antigens by genetic or biochemical approaches. Another one is development of strategies for effective presentation of tumor cells. So we have to develop strategies through which there is an effective presentation of tumor antigens by the APC cells or some other cells. So we have to develop those strategies. So now third is the generation of activated population of T cells and TC cytotoxic cells. So we have to also think about the process that which can help in the activation of TH cells and TC cells. Another process by which we can mediate the killing of cancer cell is the attack on the tumor blood supply. So what does it mean? It basically means we already know that if a tumor is present in a particular organ, then these cells are very hungry. So they have a much higher metabolism than normal cells. So to cope up with their metabolism, they start developing their own blood vessels, which is through the process, the normal process of angiogenesis they will follow. They will develop their own blood vessels, so they can get more food to develop. So the strategy which we are using here is we can attack on these tumor supply blood vessels. So if we can cut off the supply of blood or the nutrients, then these cancer cells will automatically dies. So this marks the end of today's lecture. I hope you all have liked it. If you have liked the video, then follow YTLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, and on LinkedIn. You can now the Google Play app is also present. You can download the app. So thank you for listening.